Alright, let's finish up this chapter 17, or this particular section of chapter 17, which is section 1 here. So now we want to talk about, just briefly, the genetic code here, and a little bit about this messy process of transcription and translation. So what we want to discuss is how are the instructions for assembling a amino acid chain into a protein, how is that encoded into the DNA? What is the language of the DNA? Kind of like if we talk about our own alphabet and how we can combine the letters of the alphabet to create our language and to create words, we kind of need to know the alphabet of the DNA. What is the language that this information is encoded in? There are 20 amino acids, which earlier in the semester we discussed amino acids as being part of those important macromolecules. There's 20 different amino acids, but there are only 4 nucleotide bases in DNA. So we're kind of going from a language that only has 4 letters in it, 4 nucleotide bases in DNA, and we're using that to translate into a language that has 20 letters, 20 different amino acids that can then be put together in a chain. So if you think about DNA as being a language that consists of only 4 letters, and you're translating into a language that consists of 20 letters, you kind of have to figure out some sort of pattern. How can you do that? How many nucleotides correspond to one amino acid? So that was one of the first puzzles to figure out. So you know it can't be 1-to-1, because if one nucleotide coded for one amino acid, well then that would only be 4 possible amino acids, and there's 20. So somehow we have to get to be able to code to 20 amino acids. It can't be a 2 nucleotide sequence per amino acid. In other words, it can't be 2 letters from the DNA code equal 1 amino acid, because that would only be 4 possibilities to the power of 2. That only gives us 16 codes for amino acids, and we need 20. So the smallest unit of uniform length, because we're going to find out these codes are in uniform length, that can code for all of the amino acids is a series of 3 nucleotide sequences. So again, we have a 4-letter alphabet, but we need to create 20 different words with those. And all of those words need to be the same length. Well, the only way to get that done is to create 3-letter words. And so a 3 nucleotide sequence per amino acid will get that done. That would be the 4 nucleotide sequences to the power of 3. That would give us 64 possibilities. And we only need 20, so that's more than enough. But the next step down only gave us 16. So that's the smallest we can have. We need 3 nucleotide sequences. In other words, we're going to create our amino acid words using 3 nucleotide sequences from the DNA, 3 letters from the DNA language. This flow of information from the DNA to the RNA and then eventually into an amino acid chain called a protein is based on what we call a triplet code. And of course it's called a triplet code because each one of these codes for an amino acid is basically a series of non-overlapping 3 nucleotide words. So it's taking 3 nucleotides to create this word, this language that will be translated into a particular amino acid. The words of a gene are transcribed into a complementary, non-overlapping 3 nucleotide word of messenger RNA. So we go from a gene, which is DNA, and that gets transcribed into messenger RNA. And then from messenger RNA, those words are translated into a chain of amino acids, which forms the polypeptide, which eventually would bend and fold or combine with another polypeptide to form a protein. As always, a picture is better than 1,000 words. So here, take a look. We have a DNA strand right here. It is double-stranded, of course. It's not twisted, just to clarify. But this is our DNA molecule, one long DNA molecule. This section of the DNA molecule could be a particular gene for hair color. And this section of the gene could be, or this section of DNA could be the gene for a number of fingers or something to that effect. So we're looking at this little part. See the little square? That's the tiny portion that we're looking at in gene 3. So we jump up with the arrow here. That's the section of DNA that we're going to take a look at. And this top strand is going to be our template strand. We'll discuss that word in a little bit more detail coming up. but that top strand is our template strand. And using that template strand, in this case, the top strand is, we're going to go through transcription. That's going to be our template to create a matching messenger RNA. So this is our matching messenger RNA that was created using this strand right here of DNA. Notice the only difference is instead of T, the RNA has U, but all the rest of the bases would be the same. So where in DNA you would have a T, in RNA you have uracil, a U right there. Now, this message is made up of 3-letter combinations called codons, triplet codons. So UGG, that codes for a particular amino acid. UUU, that codes for a particular amino acid. GGC codes for a particular amino acid, and on down the line. So that's the process of transcription. And that messenger RNA would leave the nucleus and go out into the cytoplasm where the ribosomes are. The ribosomes would use this as a template to tell them what to build. And so each of these triplet codons tells the ribosome, okay, build this particular protein using this amino acid here, this amino acid here, this amino acid here, and here. Put all of those amino acids together and you've built a chain of amino acids, a polypeptide, which could then be folded or changed to become a protein. So basically we're going from a language here, represented by the 2 types of nucleic acids, to a different language down here, represented by the protein. During that first transcription process, one of those 2 DNA strands, which we call the template strand, it provides the template for creating the order, the sequence of RNA nucleotides for the RNA transcript. Now, in my example, in this one, I said it was the top one is the template strand, but in other genes, it may be the bottom one. So it's not always, in this case, the top strand of DNA, but it's specific to whatever gene you're looking at. So for gene 3, the top strand was the template strand. But for gene 2, it could have been the bottom strand. It differs depending on the gene. So the template strand is always the same for that gene, but that doesn't mean that every gene uses the top strand or every gene uses the bottom strand. So for gene 3, it's always the top, but gene 2 could possibly be the bottom. During translation, the messenger RNA base triplets called codons are read in the 5' to 3' direction. So similar to what we talked about creating DNA, it kind of works in one direction from the 5' to the 3'. The messenger RNA works similar, and it reads this template in that direction from the 5' to the 3'. So you see that this set of templates is matching to the template strand up here. Those codons that make up the messenger RNA molecule, they're also read by the ribosomes, which do the translation from the 5' to 3' direction. And each of those triplets, each of those codons specifies a particular amino acid. Remember, there's 20 possible amino acids, and it tells the ribosome, okay, put this amino acid next in the chain, and then put this amino acid second in the chain, and this amino acid third in the chain, and it keeps on building that chain, creating that long polypeptide. So to create a polypeptide, which could become a protein 20 amino acids long, you would need a messenger RNA strand of 60 nucleotides, because it takes 3 nucleotides, 3 letters, to specify each amino acid. So if you have a 20 amino acid long protein that you're going to create, you need a messenger RNA strand that is 60 nucleotides long, because it takes 3 to code for each amino acid. So don't panic, this is just a brief overview. We have lots of illustrations and details to come, and we'll get some more information as we go. Before we take another break here, let's talk about cracking this code, because you can imagine how difficult it was to understand and learn the language of DNA and RNA transcription and translation. Figuring out this code, almost like breaking some sort of secret code of a war enemy or something to that effect. So all 64 possible codons, there are 64 possible codons. Remember, that's 4 to the power of 3. So a codon, remember, is a unique 3 nucleotide sequence. all 64 of these were deciphered by about the mid-1960s, so it really wasn't that long ago that we finally figured out what all of these codon messages were, basically. Of the 64 triplets, 61 of them code for particular amino acids. So 61 of those three-letter codons code for particular amino acids. 3 of the triplets are actually what we call stop signals. So 3 of the triplets do not code for a protein, but they tell the ribosome, okay, we're at the end of the message, stop right here. And the genetic code, you'll notice, is redundant because we only have 20 amino acids, but we have 61 codes for amino acids. So more than one codon can specify a particular amino acid. But the genetic code is not ambiguous. No codon specifies more than one amino acid. In other words, UUU only specifies for one particular amino acid. But there may be multiple codes that specify for that same amino acid. Codons must be read in the correct reading frame. You'll notice it might be difficult to figure this out because unlike in our sentences, unlike in our alphabet, if you read a sentence, you have capitalization and you have spacing and you have punctuation to help break up all the letters. Well, what if all the letters just ran together with no punctuation and no spacing? It might be kind of hard to figure out what the sentence said. So in these codons, there is no spacing or punctuation other than the stop codon, but you have to have the right reading frame. You have to know where to start, and you have to know that you're reading 3 letters at a time, and the cellular machinery has to know this too. So the codons must be read in the correct reading frame, the correct grouping, in order for the specified polypeptide to be produced. So think about a reading frame, again, as the correct shifting and the correct placement of letters. So here, if you want to read this sentence, the dog is brown, that's the correct reading frame for this. If we shifted by just a little bit left and right, you can use the same letters, but you create this sentence of gibberish, which means nothing. So that is basically not the correct reading frame, and you wouldn't get the information that you want. Here's what we call a codon table for messenger RNA, and you may have seen something like this before in your previous biology classes. Basically, these are the codons, so you have the first and the second and the third codon, the third letters. And if you look through here, all of these combinations of 3 codons give us a particular amino acid. They code for a particular amino acid. So notice, like, and I'm not going to recall all 20 of these amino acid names. So glycine. So look down here for glycine, which is just GLY, shortened to GLY. It can be coded for by 1, 2, 3, 4 different 3 codon letters. So if you have GGU, that codes for glycine, but so does GGC and GGA and GGG. So you notice some of them have 2 or 3 potential codes, some of them up to 4 potential codes that code for that particular amino acid. So this is kind of a table. This would help you understand the alphabet of the genetic code, kind of like a Rosetta Stone. It translates that information for you, so you can look at any of the 3 codon triplets and see what amino acid that codes for. You notice there's 3 stop codes, so UAG, UAA, and UGA are all stop codes. They don't code for an amino acid, but they tell the ribosome, oh, stop or start right there, stop right there. There's also a start code we'll look at. It codes for methionine, which is an amino acid, but it's also the start code for the chain there. So this chain, or this chart, can help you out quite a bit when you're trying to figure out that lettering system in the alphabet of the genetic code. Let's finish right here before we take another break and talk a little bit about the evolution of the genetic code. The fact that this genetic code system in DNA and messenger RNA and then into proteins, that central dogma that we talked about, the fact that that's basically nearly universal, shared by virtually all living things, from the most simple bacteria up to the most complex mammals and eukaryotic creatures. For example, the RNA codon CCG is translated, it codes for the amino acid proline in all organisms whose genetic code we've examined. So it doesn't matter if you're talking about a mouse or an oak tree or a cockroach. If the RNA codon CCG is there, that gets coded for the amino acid proline, which means that every living creature is using the same genetic alphabet to build themselves. Well, that's a strong argument and strong evidence for common descent and the theory of evolution. Even what's even weirder is because all living creatures share this alphabet, so to speak, genes can be transcribed and translated after being translated from one species to another. So you can take a gene that codes for a particular amino acid, take it out of one creature and put it in another, and it will work because it's the same alphabet. This shared genetic vocabulary, again, is strong evidence for the theory of evolution and a good reminder of the kinship of all living things. That for me, I like this idea that I and an oak tree and my dog at home and whales that swim in the ocean, that we are all interrelated, that we share something in common. For me, that's a cool feeling. So take a look at this next slide for an odd example, and then we'll take a break. So here's a tobacco plant that is expressing a firefly gene. So they took a gene from a firefly who has the ability to light up, bioluminescence, and the gene that creates that bioluminescence, they put it into the tobacco plant, and the tobacco plant grew and is able to express that gene because it's the same coding system. So no, a tobacco plant normally does not glow in the dark like this, but it can express that gene that came from a firefly, a completely different species. And here you have a pig kind of also sort of glowing in the dark here. Look at his nose and his hooves here. This pig is expressing a jellyfish gene. So again, a gene for sort of this luminescence that you sometimes see in jellyfish was put into the pig, probably as a developing embryo, and then as the pig developed into a living baby pig and then into an adult pig here, its body can express that genetics, and you see the phenotype that you would see in a jellyfish. And that works because, again, we're using the same alphabet no matter what species we're talking about. So we're going to take another break and stop right there before we get into the details. Come back and see me, and we'll get into more detail of transcription and then into translation.